My name is Kyle Uckert. I'm from the astronomy department at New Mexico State University. I'd like to start by saying today was the first time I saw rain in a very long time. It's very exciting for me. I'll be talking something a little different. Everyone else this session has talked about exoplanets, and I'll be changing topics totally by talking about planets in our solar system. First, I'd like to start by acknowledging the many people who have helped me during this project, especially my advisor, Nancy Schanover. So here's a brief overview of what I'll be talking about today. I'll talk about three types of instruments that we've been developing at NMSU and also at Goddard Space Flight Center. It's an acousto-optics tunable filter IR reflectance spectrometer, a two-step laser desorption mass spectrometer, and a LIBS instrument, a laser-induced breakdown spectrometer. And we'll be using these instruments to look at geologic samples to determine any biomineralization processes or any extant life within these minerals. And we'll be using two types of analysis techniques, a spectral unmixing technique and a principal component analysis to determine the presence of biotic activity. And we're really trying to answer this question right here. So how can biotic activity be inferred from the results of multiple instruments? So using these instruments in combination can help determine the presence of biotic activity better than any one instrument itself. So earlier we talked about biosignatures related to exoplanets, and my definition of biosignatures is a little bit different. We're looking for processes of biomineralization, or really any kind of evidence that shows that biotic activity has influenced the formation of these geologic samples. So we're looking for things like evidence of trace minerals that indicate biotic presence, evidence of a crystal structure that could indicate some kind of influence of biology on the formation of these minerals, and also any chemical or physical disequilibria with the rest of the geologic environment that could indicate the presence of biology. And the three types of instruments we're using are, as I said earlier, an IR reflectance spectrometer. So this shows us the molecular absorption features within any kind of spectra, which could indicate the presence of biology or just geologic mineralization. We're also looking at this two-step mass spectrometer, which produces a mass spectrum, which shows elemental and molecular composition of a sample, and also this LIBS instrument, which gives us an emission spectrum, which shows the elemental composition of a type of sample. So first, I'd like to talk about the AOCF point spectrometer, which has been in development since about 2009. This is an interesting kind of spectrometer. What we have is an IR light source over on this end, and after going through several optics, it reaches this AOCF crystal, which is a tellurium oxide crystal. We pulse this radio wave through at about 30 to 80 megahertz, and each time we apply some kind of radio frequency, the light passes through a certain index of refraction, which then lands on the sample and is reflected back into a detector. And after sweeping through all RF frequencies, we're able to build up a spectral image of the sample. So this particular AOCF spectrometer has a resolution of about 200 to 400, and we are looking in the 1.6 to 3.6 micron range, which is a really important range for looking for hydrated minerals or the presence of organic material. This is a non-destructive technique and requires no sample preparation at all. It's also very fast, low power, and does not take up very much space. The second type of instrument is this two-step laser desorption mass spectrometer. This is very similar to a typical mass spectrometer, but it has two lasers. So the first laser is an IR laser, which ablates some sample material from a surface. A second UV laser intersects this plasma or this ablated material, ionizing the atoms, which are then extracted into this reflectron tube. And by using the time-of-flight method, we're able to determine the mass-to-charge ratio of the particles. So we're able to determine elemental and molecular composition. And the big advantage to using this type of technique is that using two different lasers actually preserves these large, complex biomarkers much more effectively than using a single laser for ablation and ionization at the same time. This is also a very minimally destructive procedure and requires no sample preparation. So this produces about a 100 micron crater on the target. And the last type of instrument we're looking at is a LIBS instrument, which stands for laser-induced breakdown spectroscopy. And similarly, we will use a UV laser to ablate some material, and the resulting emission spectrum from the plasma is collected by a spectrometer. This is similar to the ChemCam instrument that's on Curiosity right now. The big advantage to using this type of instrument is the depth profiling. So by taking multiple shots at a sample at the same location, you're able to sort of dig into the sample and look at the very near subsurface. The type of samples we're looking at are very diverse. We're looking at first non-biologic samples, such as these sulfates, carbonates, and clays. which we think could be present on different planetary surfaces within our solar system, and also on Earth are very good hosts for life. We then dope these samples with certain biologic materials like amino acids or PAHs. And the third type of material we're looking at are just these field samples. So here's an example of desert varnish, which is plentiful down where we live. So it's some kind of substrate with this magnesium oxide crust, or manganese oxide crust, sorry. And there are microbes within this, this surface which oxidizes the manganese within the rock, which then gets excreted as this crust. And we're able to very easily spectroscopically tell the difference between these types of surfaces. And also we're looking at this which is a travertine sample with a microbial colony living within it. So here's some very brief results showing the capabilities of these instruments. This is the IR reflectance spectrometer results. And you can see very easily this evidence for hydration in these sulfates and the clay minerals. This is an example of a gypsum material which we've doped with two types of amino acids. And you can very easily see the presence of the phthalic acid and valine in both of these spectra. And here we have the laser desorption time-of-flight mass spectrometer results doped with the same types of amino acids, showing these peaks at 116 and 165 Daltons, which represent the more complex organic materials rather than just the typical elemental. So as I mentioned earlier, we're looking at two different quantitative analysis techniques to try and determine the presence of biotic activity. And although the previous slide showed good visual proofs of concepts of these instruments, having a quantitative analysis is much more thorough. So we're using two types. The first one is called the spectral mixture analysis technique. And we'll be using the USGS SpecPR process routines to apply this type of analysis to the AOCF point spectrometer. And what this basically does is it takes several spectral M members and combines them in a linear fashion in order to reproduce the reflectance spectra that we measure within the sample. The idea here is that any sample we look at is really just a sum of a mixture of different kinds of reflectance spectra. So after finding the best fit between the real and model spectra, you're able to determine the relative abundances of these different spectral M members. So there are several libraries available online which have lots of different pure samples like jarosite or sulfates. And we're also going to add to these spectral M member libraries different types of organic materials and lichens that we'll be trying to observe. The second type of analysis is called principal component analysis. I'll just give a very brief overview of what that is. So this will be applied to all three instruments using a commercial software package called the Unscrambler. And the principal components are really defined as the dimensions within a sample set along which the least amount of variance exists. So this is a really simple example showing a two-dimensional data set, which is just very ambiguous. And it shows that the first principal component would be along this axis, and the second would be along this axis. So this is only a 2D data set. With the LIBS instrument, for example, we're looking at a 13,000 dimensional data set, which is much harder to visualize, but the principle is really the same. Now, this allows you to determine what the relative quantities of trace elements within a sample is. It could also help you determine what the method of formation is based on the amount of energy that's necessary to ablate a sample. And it also is able to infer the biologic content of an unknown sample. So I want to show an example that we've taken very recently. This is a very blurry picture of a monzonite sample with lichen on top of it. So I've only shown the picture of the lichen, but we're essentially looking at three different regions of this sample. We have a very large rock which has this lichen crust on it, and you can see that there are these green areas, some black areas, and then not shown in the picture is the cut surface. We've cut with the rock saw, so this is a very smooth surface, and a crust without any lichen at all. And when we apply the PCA technique to this, we see that the different types of surfaces we observe break it out into these four different groups. So down here we have the black lichen area. Over here we have the green lichen area. Up here is the crust without any lichen that's visibly observable, and over here is the cut rock surface. So it's difficult to just see what this really means unless we look at what are called the loading plots, which show how the variance is actually defined. And in this case, principal component 1 is really based on the amount of silicon in the sample. And we see that the areas with lichen have much more silicon in them than the areas without. Lots of lichens produce a silica glaze over top of itself as a way of, they think, protecting it from the harmful UV rays. So that could be what we're observing here. And also along principal component 2 is an increase in magnesium and carbon. And that's shown that the lichen has more magnesium and carbon. We're not quite sure what this could be, but it's possible that this could indicate the presence of chlorophyll or chloroplasts, which we're observing here. So the question we're trying to figure out now is how to apply these results to all 3 instruments. And for future acquisition, what we plan to do is take in situ measurements in cave regions. So in October, we'll be taking the AOCF point spectrometer and the LIBS instrument into Snowy River Cave. So this is in Fort Stanton, New Mexico. And it's known that many different kinds of the speleothems or cave formations within this cave are produced through biotic means. So they're influenced through microbial activity. And we'll be trying to use these analysis techniques to determine which types of cave formations are produced through biology and which types are produced through abiotic mechanisms. And the whole point to this, or the astrobiological context to this, is we're trying to determine how each of these instruments can be used to look for life in the solar system, in that if you are planning a roving or landing mission to another solar system body and you have an idea of what kinds of life you're able to find, we'll be able to tell you which types of instruments or multiple types of instruments are best suited to observe those kinds of biologic mechanisms. And that's it. Take any questions. Thank you. Yes.
How big does the mineral have to be in order to get a reading?
The question was, how big does the mineral need to be? The AOTF basically produces that size limit, and it has a spot size of about 1 millimeter in diameter. So anything about twice as big as that is usually pretty good for us.
So you usually try to use a pure mineral?
To start with, to build the spectral M-member library, we use the pure minerals, and we're starting on using these mixtures or field samples. Yes?
For the organics you were discussing about analyzing, you're talking about peptides and that sort of stuff. Do you think you're pushing the lipids, anything like that?
It's not something we thought about, but it's something maybe we should look into. And I know those are usually used for semi-permeable membranes, I think. So that's definitely a good indicator for life. Yes?
Do you know the different isotopes of carbon?
We're not able to measure the different isotopic ratios for carbon. The laser desorption time-of-flight mass spectrometer might, but the signal to noise just isn't good enough for us to do that. Yes?
Which amino acids you spike your samples with and why you
Chose those in particular?
can you read that?
Yeah. Can you repeat what you said about which amino acids you spiked your samples with and why you chose those particular amino acids?
Sure. We chose phthalic acid and valine, and I think they were chosen because those were the ones we had available. Sorry.
Can you distinguish between the different isomers of valine with your methods?
Sorry, I don't think I can answer that for you. Okay, thank you.